Mendelian Inheritance. So, Gregor Mendel, this fellow here, was a monk who essentially figured out how traits are inherited and discovered the idea that these traits can be carried in sort of pairs called alleles. So he worked with pea plants to figure this all out, and again, essentially came up with the laws of inheritance. And he's really big in using statistics, or you're essentially applying mathematics to his observations to look at the probabilities of certain things happening within inheritance. So why the pea plant? He chose the pea plant because it is really easy control, essentially. It can self-pollinate or cross-fertilization, where you have two different plants pollinating each other. And so you can very easily and relatively quickly control what traits a group of plants is going to have. It's easy to work with. So if we look at the structures, they're easy to basically manipulate, so they will only breed with whom you want them to breed, and their characteristics are easy to distinguish between as well. So they have contrasting traits, of which there's a limited variety, only two possible outcomes. So for example, the seed pods themselves could be differently shaped, and so he used different terminology to describe them. The flowers could be purple or white. The height could be tall or short. The seed texture, if we look at these ones here, so it could be like a round pea or a wrinkled pea. The seed color itself, the color of the pods, and so essentially, and there's the color of the flowers. Essentially, there was a number of traits that there were two varieties of, and very easy to distinguish between those two varieties. So using the word characteristic versus trait. So a specific characteristic will have possible traits. And so example, the flower color is the characteristic. And so when I mention the word variety, the proper word for that is that the characteristic of flower color has two possible traits. And so the options are the traits, and the thing that is applying to, so the color of the flower, is the characteristic. So Mendel's experiment, essentially he created purebred plants whom, if you looked at the group of them, anytime they reproduce, they would create only one trait of that particular characteristic. So for example, they would always produce tall plants, and all their offspring would also be tall plants as well. So we would see only one version of the trait for a particular characteristic. He would then cross-pollinate these purebred plants with different traits of a particular characteristic. So if we talked about the height being the characteristic, he could breed one plant with a tall trait and one plant with a dwarf trait, and then see what happens, and essentially look at the numbers produced and figure out how these traits are inherited. So the purebred plants are referred to as a P generation, so that's the parent generation. And then the offspring, which are going to be a hybrid between the two crosses, are the F1 generation. So the F1 generation, these are called monohybrids, and this is where they're going to have a hybrid of only one characteristic. So we're going to start off by looking at monohybrids, but then also we'll look at dihybrids as well. So a monohybrid is only one particular characteristic that we're looking at. So we could look at height, or we could look at flower color, but we're not going to look at both of them at the same time while we're doing monohybrids. When we do a dihybrid, we'll look at two of them at the same time. So this type of cross is called a monohybrid cross, where we're taking only one of the characteristics. We're looking at two different traits of that characteristic, and we're going to cross them. So that's called a monohybrid cross. So one experiment, we can look at making an F1 generation from, as we said earlier, a tall plant crossbred with a dwarf plant. So that's the parent generation. And so Mendel would do this, and then he'd get the seeds of them, and he'd plant the seeds, and then look at the phenotypes, or essentially how the traits were expressed of the next generation, the F1 generation. And so essentially when he did this experiment, you cross a tall plant with a dwarf plant. What he saw was that the F1 generation, all of them were tall. So if there were 4 offspring, all would be tall. If there was 1,000 offspring, all of them will be tall. We'll look at why there's specifically 4 here, but we're going to essentially lower it down to the lowest whole number ratio. So, results, all the F1 plants are tall. Then, if you take that F1 generation, which is a tall plant, and you take another member of that generation, another tall plant, and you cross them again. So he's taking the F1 generation and crossing 2 of those plants to form a third generation, which would be called the F2 generation. What he found was that 3 of them were tall and 1 of them was a dwarf plant. So that trait of the dwarf height in these plants was still present even though it wasn't shown in the F1 generation. When these F1 generations were bred, they created mostly tall plants, but they also created some short plants, and the ratio ended up being 3 tall to 1 dwarf plant. And so he did this with these traits and kept track of the numbers to figure out how it is that these traits are inherited. So the results, 3 tall, 1 dwarf plant. One of the reasons why he was successful as a scientist by doing this is not only did he just breed pea plants together, but he did many trials. And so essentially every trial that you do is going to have some error to it. You do many of them, and you can start to minimize those errors. Keeping detailed records, so he wasn't just a gardener planting pea plants and saying, oh look, that's interesting. He actually kept detailed records of it and tracked everything. And then really importantly, he essentially used statistics to analyze the data. Okay, how many plants are there of each type? And from that, he was able to draw his conclusions. So here are some of the characteristics. And so if we look at the parent generation, if you cross a round with a wrinkled, all of the F1 generation are round, but then the F2 generation, most of them are round, but some of them are wrinkled. And then same thing with seed color. If you look at the green and the yellow, all of the F1 generation ends up with yellow peas, but the F2 generation, the third cross, so there's one cross that produces the F1, another cross that produces the F2. If you look at the numbers again, there's more of these yellow ones and fewer of the green ones. And so looking at each of these characteristics, he essentially planted all sorts of pea plants and did the crossing of the parent generation to produce an F1 generation, and then crossed 2 members of the F1 generation to produce the F2 generation, and then kept track of the numbers for each one. And these numbers here, essentially they're large numbers. It's sort of hard to see the ratio, but if you reduce them to the nearest whole number, you can get an idea of how these traits are passed down and inherited. So Mendel's conclusion was that for each characteristic, so the color of the flowers or the height of the plant, there were a pair of alleles, which he didn't call alleles at the time, but we were going to call them alleles now. And a plant could have, so it's got 2 versions of these alleles, it's got a pair of these alleles, and they could be identical or they could be different. And from what we've seen by looking at mitosis and meiosis, we know that these pair of alleles, they essentially are the chromosomes. And so if you have 2 versions of the same chromosome, so let's say this is chromosome number 4, you would have a maternal one. Oh, let's change that a little bit better sized. A maternal one and a paternal one, of which contains the same information. So you have 1 version and you have 2 versions. These are the factors that Mendel was seeing interact in the breeding of these plants. And so we are going to look at these alleles, and essentially what that is that on this chromosome here, you have certain genes on there that code for something like, let's say, flower color. So you have one on one chromosome, and you also have the same gene on the other one. Now those genes could be the same, identical, or they could be different. So for example, the purebred generations, they would have the same chromosome, the same trait, or what we're going to start talking about is the same allele for that particular characteristic on both of its chromosomes. But when we get into the F1 generation, what we could see is that even if you had one parent that was pure purple flowers, another parent that was pure white flowers, they would only have identical versions. But when you cross those two, you could end up with, let's put a different color in here, you could end up with this particular trait having two versions, one on one chromosome and one on the other chromosome. So one allele could express itself and be dominant over top of another one. So let's say this was seed color. And so there are two traits for seed color, yellow or green. And so if a plant ended up with, let's actually do that. Let's say a yellow version and a green version for the gene that encodes seed color. So if that individual had both of those alleles, both of those genes, one on one chromosome, one on the other, he saw that one of them could be dominant over top of the other one. So even though this plant might have a gene for yellow and green, it ends up having yellow seeds because the yellow trait could be dominant. The other one gets the term recessive. It's there, but you don't see it showing up in the makeup of the plant, the phenotype of the plant, how those genes are expressed. So this is the law of segregation. An organism can carry two alleles for each characteristic, one from each parent. So think of that as the homologous chromosomes. A pair of alleles is segregated, separated, when a gamete is formed. And so we saw during meiosis that those homologous partners are going to separate from each other. And so we end up with those two traits being separated in the formation of the gametes. And therefore, the chances of the offspring getting them are going to be a particular number as well. So, as we mentioned, phenotype is the observable characteristic of an organism. So its genes are there with the information, and we refer to that as the genotype. But when they are read by the cells, they become certain things like seed color or flower color or wrinkled seeds or non-wrinkled seeds. What they actually become, the observable characteristics, that is the phenotype. The genotype is the genetic information itself. So we're going to talk about what genotypes they have, and the genotype dictates what the phenotype is. But remember, because certain traits can be dominant and certain traits or certain versions can be recessive, the genotype may not completely be observable in the phenotype. And what I mean by that is that a plant could have genes for both a purple and a white flower, but it will only express one of those two because the traits themselves could be dominant or recessive. So alleles are the variations of the same gene, and so for this particular one, we're going to be using letters to make this a little bit easier to talk about. If the trait is a dominant one, we're going to say that we'll use a capital letter, and if it is a recessive one, we're going to use a lowercase letter. And essentially, these are for a particular characteristic. And so we can choose the letters based on that as well, but the dominant one capital, the recessive one little one, and those represent the alleles. So we have, in this case, we have the dominant allele here, and we also have a recessive allele for this particular characteristic of height. Homozygous is when the genotype is the same for both alleles. So you could have two tall alleles, or you could have two dwarf alleles. Both of those are they're the same within the same individual. So this individual here has two tall alleles. This individual here would have two short alleles. That's referred as being homozygous. Then, if you have one of each, we refer to that as heterozygous. And so if you have a plant with one tall allele and one short allele, that plant's genotype could be referred to as heterozygous. And now the dominant one, if the individual is heterozygous, the dominant one will be expressed over the recessive one. So its phenotype will be expressed as the dominant of the two. A Punnett square is what we're going to be using to keep track of these. And so essentially we're going to look at the parental alleles, and we're going to say, okay, well, if the parental alleles are whatever they are, we can get these possible combinations between the two parents. So we're looking at the probabilities of certain genotypes showing up in the offspring. And we're going to organize that as a table or a grid, and then we'll be able to look at what the possible outcomes are. So example, a homozygous green plant. And so we're going to use G to talk about this particular characteristic. We're going to use a capital G to talk about the dominant trait of this characteristic, and we're going to use little g's to talk about the recessive version of this characteristic. So a homozygous green plant will have two capital G's as its alleles. So you can think of those, it's a gene that's on the chromosomes, and this particular one is saying that a homozygous green plant has one gene for making it green, green peas, and the other one also making it green. And this is crossed with a homozygous yellow pea plant. And so we're going to cross this with another plant that has, it's also homozygous, but it's homozygous yellow. And so it has the two alleles that would allow its peas to be yellow. So two lowercase g's will represent the two genes that allow this plant to produce yellow fruit. Now the question is why not use its yellow, so why not use y's, right? And the reason is that we want to look at the same characteristic, and so the characteristic we're looking at here is the color of the peas. And because we're looking at that particular characteristic, we want them to be the same letter, in this case G's. We just want to have two versions of that letter. And so the characteristic tells you which letter you're dealing with. The trait makes it either a capital or a lowercase, depending on whether it is a dominant one or a recessive one. And so if we cross these two, we could say, okay, one parent is homozygous with two capital G's. We can put them on our table and say, okay, this here represents the parent that is the green pea plant. So let's make that actually green. So this parent has two alleles, giving it green peas. Then we want to put up the parent number 2, which is a yellow pea plant. And again, we're not using Y's because we're talking about the same characteristics. So we want to stick to the same letter, but it's homozygous recessive, and that's going to make it yellow. So we want to put them on the grid as well and say, okay, here is the other parent, and we'll space them out like this so we can actually get a grid. This is the yellow pea bearing yellow pea plant bearing parent, and this other one is the green pea plant bearing parent. And so we can now look at the possibilities that could happen when the green parent combines with the yellow parent. One thing that could happen is that let's look at the combinations here. If this gene ends up in the sex cell, and this gene from this other parent ends up in the sex cell, if they come together and form a zygote, the genotype of that zygote would be capital G, little g. But the yellow pea plant parent has two possible alleles, and so the other one could end up in the sex cell as well. And if it was used with the capital G from the green parent, we would end up also with another capital G, little g, forming the zygote. Same thing here, if the capital G is passed on, if it's forms the sex cells that form the zygote, and the little g is passed on, we end up with capital G, little g, and the last combination would be capital G, little g, as well as we use both of those zygotes. And we do tend to put the capital letter first, not that it would make too much of a difference. But what this means is you would end up with 4 offspring. If this is the combinations that you're using here, you'd end up with essentially 4 offspring, all of which would have the dominant green gene being expressed in the phenotype. So you would end up with 4 offspring that are all going to produce green peas. And they are going to be not homozygous offspring, they're going to be heterozygous offspring. So we get 4 heterozygous offspring with capital G, little g, and their phenotype would be all of them produce green pea plants. We get none that produce yellow pea plants. They are all producing green pea plants. All right, realize that these 4 here that we're talking about, offspring, not every time plants reproduce do they produce exactly 4 offspring. This would be a probability. And so essentially you could think of it as any multiple of 4 would be relatively easy to do. So if they created 400 offspring, let's say, all 400 of them would have heterozygous genotypes, and all 400 of them would produce green peas. So think of this as a ratio that we're creating, and you could easily scale up that ratio to any number you want. All right, let's look at number 2. With a heterozygous purple flower plant is crossed with a white flower plant. Complete the Punnett square to show the cross and state the genotype and the phenotypic ratio, so the outcomes of them. So the first one is we have a heterozygous purple flowered plant. And then we're going to be crossing that with a homozygous white flowered plant. And so if the flower is purple, but we know that it is a heterozygous purple plant, then we can say, okay, well, let's talk about this, this characteristic is the flower color, of which we have 2 traits, and let's use the letter P, which is not great because capital P and little p kind of look the same when they're not beside each other, but let's use P as a capital. That represents the particular characteristic that we're dealing with is flower color here, and the dominant one we know is the purple one, because if it's heterozygous and it's purple, then purple must be dominant. Heterozygous means it also has a recessive allele. We have the dominant purple allele, and then we have the recessive allele, which we know the other option is having a white flower in this case, and so the recessive little lowercase p is representing a white flowered allele. So therefore, if the other parent is homozygous white, we would use little p to express their genotype. Now, again, we're using p's. We wouldn't use w's in this case because we want them to have the same letters so that we can look at the genotypes when they come together. So again, for any given characteristic, make sure you're using the same letters. Okay, so let's put them up here. We have our, let's do the purple one here, purple flowered plant, and we have the white flowered plant. And again, each one of them is going to contribute a allele to the zygote. And so if you use a capital P and a little p, we're going to combine to form a capital P, little p zygote. In this case here, we're using 2 lowercase p's, or we're combining 2 lowercase p's. So this particular zygote would have 2 recessive alleles. This one here, we got the capital and lowercase again, and this one here, we have 2 recessive alleles. So if we look at the genotypes, there are 2 of them that have the heterozygous traits, where they have 1 uppercase p and 1 lowercase p, and then we have 2 here that are homozygous for the recessive trait, the lowercase p. So we can write that out and we can say, okay, the genotype is we get 2 of these heterozygous purple plants, and technically when we say purple, that's the phenotype, and then we have 2 of these homozygous genotypes as well, which is both of the recessive traits. The phenotypes would be that these guys here, because they have a dominant purple allele, they are going to make purple flowers. These guys here, these 2 right here, we have 2 lowercase homozygous recessive traits, so these are going to produce the white flower plants. And so we have the genotype and the phenotypes based on who the 2 parents are. So we use these Punnett squares to essentially look at what are the possible combinations when these 2 genotypes of the parents are going to be bred. And again, it is just a ratio. So when we say 2 and 2, that's the lowest whole number ratio. But if you bred 500 of these plants, you would essentially end up with half of them being purple which would be heterozygous, and half of them would be white, which would be homozygous. So if you bred 500 of them, you get 250 purple ones and 250 white ones.